PathLit-Agent is an AI system for automated biomedical literature mining with a focus on pathways. I will walk you through the motivation behind this work, the system design, and how PathLit-Agent supports efficient discovery in bioinformatics and disease research. Biological pathways represent how genes, proteins, and molecules interact to carry out functions within a cell. They allow us to move beyond single gene observations and understand coordinated biological behavior. When pathways are dysregulated, they often drive disease progression, drug response, or therapeutic resistance. Studying pathways gives us a systems-level view. The main challenge is that pathway-focused literature review is very time-intensive. Manually searching and reading papers doesn't scale, especially when hundreds of abstracts need to be screened before useful information is found. Even after reading, extracting genes, interactions, and mechanisms is slow and repetitive. Generating a structured summary or report for just one pathway can take hours, making it inefficient for large-scale research. This is the execution flow of PathLit-Agent. When a user submits a query, the system first checks memory to see if a similar pathway report already exists. If a high similarity match is found, the cached report is evaluated. If gene coverage is acceptable, the Markdown report is returned immediately. If not, the system refines the query and reruns the pipeline to improve coverage. If no suitable memory match exists for the input query, PathLit-Agent runs the full workflow. In Phase 1, it plans PubMed queries, retrieves papers, and screens them for relevance. This step operates as a loop agent, repeating until a reliable paper set is collected and passed forward as structured JSON. In Phase 2, the agent extracts pathway entities such as genes, interactions, and mechanisms. A summarization loop then generates and refines summaries, checking consistency as it progresses. In Phase 3, the final structured Markdown report is constructed. The report is then automatically evaluated, refined if necessary, and finally stored in memory, allowing future queries to be answered faster and with higher quality. This is a short demo to show PathLit-Agent in action. Here I am entering the query, role of PI3K ACT in breast cancer, into the PathLit-Agent interface. Once submitted, the system begins running the multi-stage pipeline. Because this is the first time the query is seen, we need to wait while the agents retrieve papers, extract entities, summarize the mechanisms, and construct the report. In the lower panel, you can see the back-end terminal output, showing the PubMed retrieval steps, entity extraction, summarization loops, and report evaluation happening in real time. When the pipeline completes, we copy the generated Markdown output and paste it into a Markdown viewer for better readability. The summary section and gene evidence table are automatically produced from the extracted literature. Now, when we run the exact same query again, the response is almost instant. The previous result was saved into memory, so instead of rerunning the entire workflow, PathLit-Agent retrieves the cached report and returns it immediately. PathLit is powered by Google ADK agents, Gemini reasoning models, Europe PMC retrieval, Gradio Lite UI, and a fast API backend. We store embeddings in SQLite for long-term memory and rapid recall. PathLit-Agent is still a newcomer, a system with room to grow and improve. That was PathLit-Agent, still early, still improving, but already useful. Thank you.